Hello, Dr. Debbie Morris here, and today we are going to continue talking
Presbyopia
about the
develops when our eye's natural
physiology of
lens
the
loses
nervous
its
system.
flexibility. Focusing
And
up close
we're
becomes
going
more
to
difficult.
focus
The eye's
primarily
ciliary
in
muscle
this lecture,
contracts,
which
which causes
again
our
will
flexible
probably
lens to thicken or
be
change
divided
shape.
into two videos
This
because it's
change in shape
very
allows us
dense
to
and
see
very long.
objects up close. As we
We're going to be
age,
talking
our
about
lens
special
becomes
senses,
less
about
flexible
the
and
sense of vision,
resists changing
the
shape,
sense of
so our
hearing,
ability to see up
and
close is
the
diminished.
vestibular
Usually, the
system, which
first sign of
provides us
presbyopia is the need to
with
hold a newspaper or
balance
other
and
reading
a
material
sense of
at
position
arm's
in
length to be able to
space.
read them.
So we'll start with a little bit of just general neurophysiology, talking about something called glia. And glia are cell types in the central and peripheral nervous system that are not neurons but are integral to the nervous system and provide important support mechanisms to the neurons and in general to the nervous system. So we earlier mentioned something called Schwann cells that make the myelin sheath in the peripheral nervous system. There is a kind of cell in the central nervous system called oligodendrocytes. They're a little bit different, but they both wrap around axons to provide a lipid insulation to allow for more rapid conduction of impulses. There are glia that provide nutrition to cells, that have immune functions. Glia in general are really helpful in maintaining homeostasis. There are glial cells that make cerebrospinal fluid, which cushions the brain and the spinal cord, and which provides some hormone transport for the hormone melatonin and also some nutritional support to the central nervous system. There are cells that create what's called the blood-brain barrier by wrapping around capillaries and preventing certain substances from easily crossing into the nervous system where they might be toxic or have adverse effects. And then glia, as we learn more about them, we have found that they're actually really important in helping to form and maintain neural networks by assisting neurons in forming synapses and connections. This slide just is a diagram of some of the types of glial cells, most of the types of glial cells in the nervous system. You can see these are intended to be neurons, so they have a cell body, dendrites, and axons. In the central nervous system, oligodendrocytes have processes that wrap multiple times around axons, creating the myelin sheath. This is a little different from Schwann cells. I don't know if you remember from the diagrams, but when we were talking about peripheral nerves, single Schwann cells with their nucleus wrap multiple times around a single axon. And there are multiple Schwann cells along the length of the axon. Oligodendrocytes, on the other hand, send out their processes to multiple axons, and there are multiple oligodendrocytes along an axon, and there are multiple axons that are attended to by given oligodendrocytes. And you can see there's some communication here between the glia, various types of glia, and the neurons. Astrocytes have a number of functions, and one of them is creating this blood-brain barrier by being actively involved with capillaries. Ependymal cells line the spaces that contain cerebrospinal fluid, and they circulate cerebrospinal fluid, and some of them actually make cerebrospinal fluid in certain places. And then microglial cells, which have immune functions. So here in the peripheral nervous system, we have Schwann cells acting to make the myelin sheath, and something called satellite cells that basically are in close proximity to the neurons in the peripheral nervous system, for instance, in the dorsal nerve root, where the primary somatosensory cells are, and they help to support the cell bodies. Something in addition glial cells do is that they make growth factors that help to promote the health and growth of neurons, and in fact, the sometimes reproduction of neurons. It's not true that you can't grow new neurons. It's used to be true, but we've learned more. It's hard, but there are important growth factors that are made by some glial cells, particularly by Schwann cells and astrocytes. Again, in the central nervous system, the oligodendrocytes make the myelin sheath. Astrocytes have a ton of functions. So they provide support for the central nervous system in terms of nutrition, among other things. They are part of the blood-brain barrier, again, wrapping around the capillaries to help prevent easy transfer of substances from the vasculature into the nervous system. They make growth factors and neurotrophic factors, and they are also involved in absorbing excess potassium in the extracellular fluid, which of course we know all cells are doing because of the sodium potassium pump, but also they take up neurotransmitters. So used neurotransmitters, some of them may be broken down by enzymes, some of them may be retaken up into cells, but some of them are taken up by astrocytes just for recycling of their components. The microglia are immune cells and they act as scavengers. They basically help to clean up messes. They can move, so they have the ability to move through the nervous system in some cases, and they are phagocytic. And then finally, the ependymal cells, which line the compartments within the central nervous system that contain cerebrospinal fluid, and they are ciliated. On that, those cilia help to move the CSF. This is the same thing again. I believe astrocytes help to form scar tissue, so they have some connective tissue function. All of them, many of them help to provide structural support. And there's not a whole lot new there, except that the microglia are the macrophages. Macrophages being immune cells throughout the organism, but the microglia are the macrophages in the central nervous system. Before we move on to vision and the special senses, I want to refine something that I kind of briefly mentioned in the previous lecture, because this is an important clinical concept to help you understand injury and damage in the central and peripheral nervous system. There is a division between types of motor neurons. There are upper motor neurons that are located in the brain, either in the cerebral motor cortex for most of the body, or in the brainstem for the motor function of cranial nerves, like the seventh cranial nerve that moves the face, and other cranial nerves that have to do with movement of the pharynx and the tongue. Upper motor neurons, so they originate in the central nervous system. The axons cross from the left to the right or from the right to the left in the medulla, and then they travel through those descending motor pathways we saw at the end of the last lecture, the lateral corticospinal tract. and they synapse in the spinal cord, so still in the central nervous system, with lower motor neurons. When damage occurs to upper motor neurons, it tends to be things like strokes, causing cell death in the cerebral cortex, head trauma and bleeding, again, causing damage in the cortex, and birth ischemia. So, people with the neuromuscular effects of cerebral palsy have upper motor neuron damage. And when upper motor neurons are damaged, you get weakness because you have lack of input to the relevant muscles, but you get spasticity or stiffness, hyperreflexia, so very brisk overactive reflexes, something called clonus, which is a rhythmic jerking when a reflex is checked, and a positive plantar reflex or Babinski sign, and that's obviously if it is damage to upper motor neurons that innervate the lower extremity. The lower motor neurons are located in the spinal column, in the anterior gray column, or some of them are in the peripheral nervous system in the anterior nerve roots. And then the lower motor neurons that synapse with upper motor neurons that were originating in the brain stem. The lower motor neurons are also in the brain stem, in this case in the cranial nerve nuclei. And the axons of the lower motor neurons extend out to the muscles, to the voluntary skeletal muscles. When a lower motor neuron is damaged, it tends to be by events such as spinal cord injury. So if you break your back and have damage to the spinal cord, that is lower motor neuron damage. If you actually cut or crush a nerve originating from the spine, so say you have a deep laceration in the leg that severs the femoral nerve, that is a lower motor neuron injury, which denervates or takes away the nerve input to skeletal muscle beyond the injury. And when lower motor neurons are damaged, the symptoms are weakness or paralysis, but it is not spastic stiff paralysis, it is flaccid floppy paralysis. Reflexes become absent or decreased, not hyperreflexic. The muscle atrophies, and sometimes you get something called fasciculations, which is uncoordinated contraction of the muscle, making it look like sort of a bag of worms. And this is a chart just comparing the signs and symptoms of upper motor neuron damage and lower motor neuron damage in a disease called amyotrophic lateral sclerosis. And in ALS, you can have both upper motor and lower motor neuron damage. The upper motor neuron signs, which are things that you detect on exam, include spasticity or tightness of the muscles, a slowing of the ability to do rapid alternating movements, an increase in reflexes, and disorders of gait that have to do with the spasticity. Upper motor neuron symptoms, things that people complain about, are stiffness and slowness and lack of coordination, spontaneous clonus, so this sort of rhythmic jerking when a muscle is stimulated, spasms of the flexor muscles causing contractions. And then they compare to lower motor neuron signs and symptoms. On exam, a lower motor neuron lesion would result in weakness, in reduced reflexes, in muscle atrophy and fasciculations, and in gait disorders related to the weakness. And The symptoms, the things people complain about, are weakness and muscle atrophy and fasciculations and sometimes cramps. I want to move on now to the special sense of vision, which is really interesting, really crucial to the ways in which we interact with the world. And the organ of vision is the eye. And the eye is a very complicated structure, so it's important to review the anatomy of the eye and to understand how the eye is constructed to help us understand how it works. The eyeball consists of a fibrous outer layer. The part of which we can see is called the sclera. Okay, it is covered with a mucous membrane called the conjunctiva, and the cornea, or the clear portion in front of the iris and lens and pupil, is part of that fibrous outer layer. So that's the eye. There is a middle layer, which is vascular and contains tons and tons of blood vessels, along with connective tissue. And then the innermost layer of much of the eye is a neural layer called the retina. And we will talk more about the retina and its layers and the kinds of neurons in it as we move through this lecture. There's a clear gel that fills the globe that's called the vitreous. And then there are some really important structures anteriorly. Um, there is a fluid filled space called the anterior chamber in front of the iris and pupil. There is a fluid filled space behind the iris and pupil that is called the posterior chamber, and the fluid in there is continuous, runs between them. There is a clear lens, which is quite flexible in youth, becomes less flexible in age, and which assists the cornea in focusing light on the retina. And we'll talk a little bit more about the iris and these things called the ciliary body and the ciliary muscle as we move through this lecture. You can see that the retina, being formed of neural layers, there are axons that are traveling from neurons in the retina, which collect at the optic nerve and leave the eye to enter the central nervous system. There is a part of the retina where light detectors are most dense and where vision is most detailed, and that is called the macula. So just a review of those same things. We see the sclera and then the vascular middle layer, which is called the choroid, and then the retina, which is composed of neurons. The anterior structures, including the cornea, which is continuous with the sclera, the anterior chamber filled with fluid, the iris, and then the pupil, which is the opening in the center of the iris, this clear lens, okay, the ciliary body and muscle attached to the iris and to the lens, actually attached to the lens, sorry, not to the iris. The vitreous gel, the macula, which is the central, it's a little depression on the retina, but it is where um, uh, cones are most dense. And then the optic nerve, and the blood vessels to the eye come in through the optic nerve. There are also exterior structures. We have an upper and lower eyelid. Um, There are eyelashes that help to protect the eye. Medially, we have little holes that are called tear ducts that allow the tears to leave. The tear gland is actually laterally in the upper lid. And and then of course there are muscles that are attached to the globe of the eye, and those are what we call the extraocular motor muscles. They include cranial nerves 3, 4, and 6. And you'll learn a lot more about them as you move forward through physical exam, for instance, and anatomy. It's important to understand the aqueous humor, which is this watery fluid in the anterior and posterior chambers, so surrounding the iris and in touch with the lens. It is continuously secreted by cells that line something called the ciliary body. So there is an epithelial lining of the ciliary body that's continuously secreting a little bit of aqueous humor. So 2 to 3 microliters per minute. Not a lot, a little bit. And then that fluid migrates from the posterior chamber between the ligaments that support the lens and then through the pupil into the anterior chamber. And it leaves the anterior chamber as continuously as it's being produced. It has to be taken away or you'd have an increase in pressure. So it flows into the angle between the cornea and the iris and through something called the trabecular meshwork to enter the canal of Schlemm. And the canal of Schlemm empties the fluid into the veins around the eye. And here we have a picture. So this is the anterior chamber, the posterior chamber, the processes of the ciliary body. So the ciliary body and the ciliary muscle coexist here. The ciliary muscle has ligaments that suspend the lens. The ciliary body is producing very continuously and slowly the aqueous humor, which then flows out between the iris and the lens into the anterior chamber and then leaves the eye through this thing called the trabecular meshwork, entering Schlemm's canal and finally emptying into extraocular veins. Why is it important? Well, I mentioned that if you are making aqueous humor and you are not constantly getting rid of the aqueous humor that you make, you're going to get an increase in pressure in the anterior chamber. So here we have a case, and that's a pretty scary looking eyeball there, isn't it? 72-year-old woman presents to the emergency room complaining of severe bilateral eye pain, vision loss, nausea, and vomiting. And when you get a little more history, she says she had a cold and she took some Sudafed and some NyQuil a few hours ago, and the pain started after that. So, one of the things that we need to know clinically is what are these drugs, right? Sudafed is a decongestant and it's a sympathomimetic. In addition to being a Sudafed being a sympathomimetic, NyQuil usually has most formulations of NyQuil have some decongestant in them, which are sympathomimetics. So here we have a little bit of a double dose of sympathomimetics. And what do sympathomimetics do to the pupil? Do you remember? What does epinephrine? What does epinephrine do to the pupil? It makes it dilate. Now, NyQuil usually also has antihistamines in it because they help people sleep. And antihistamines have anticholinergic effects, like atropine, like belladonna. And what do anticholinergics do to the pupil? Well, they also dilate the pupil. So those drugs dilated the pupil. And what happens in a specific kind of glaucoma called narrow angle glaucoma or closed angle glaucoma is that when the pupil dilates, it blocks the ability of the aqueous humor that is formed in the ciliary body to pass through the trabecular network into the canal of Schlemm, building up pressure. And that pressure causes, you can see the dilated pupil, you can see some haziness or fogginess of the cornea. You can see that the eye is red, so this is a cause of red eye, and it causes really extreme pain. A little bit more about the iris and the ciliary body. The ciliary body also contains muscle, and that muscle is attached via something that are called zonules or suspensory ligaments to the lens, which is this clear, relatively flexible structure behind the pupil. Ciliary body is also continuous with the iris. Now, the ciliary body, as I said, includes both muscle and an epithelium that makes aqueous humor. The muscle, and this is one of those more difficult things that I've actually gotten confused about a number of times, so I hope I don't get confused as I'm trying to teach you about it. But these muscles that attach this to the lens via the zonules or suspensory ligaments, that's called the ciliary muscle. And when the ciliary muscle contracts and relaxes, it changes the shape of the lens. And by changing the shape of the lens, it changes essentially the focal length of the eyeball and changes how images are focused on the retina. So there is a reflex called accommodation. When you look far away, your pupils are more dilated and your lens is pretty flat and you're kind of relaxed because your ciliary muscle is relaxed. When you change your focus from something all the way over there to something right in front of your eyes, your pupils constrict a little bit. Your lens becomes rounder, thicker, more convex. And the muscles of your extraocular muscles verge. They make you adduct your eyes to look together. When there's no tension on the capsule of the lens, which happens when the ciliary muscles are relaxed, which happens when the ciliary muscle contracts. The lens relaxes when the ciliary muscle contracts. When the lens reacts, it rounds up. It becomes kind of almost spherical, very convex. And when the ciliary muscle becomes inactive or relaxes, then the zonule fibers tense and flatten out the lens. And that's why, and it's kind of counterintuitive that the lens tenses when the ciliary muscle relaxes with accommodation, and the lens relaxes when the ciliary muscle uh, tenses and the lens becomes rounder, and the ciliary muscle tensing releases the pressure on the lens. And so it doesn't take energy particularly to look far. So far vision doesn't tend to cause the kind of eye strain that near vision causes. When you're looking near, um, and the ciliary muscle is contracted and tension on the lens is released and the lens becomes round, that uses more energy and creates more fatigue than looking far away. So here we have a picture of the iris, showing the ciliary muscles relaxing, and that allows, when the ciliary muscle relaxes, that pulls the suspensory ligaments and flattens out the lens, and you look far away, you focus far away. But when you focus on something close, the ciliary muscle contracts, releasing tension on the lens, and the lens becomes more globular. And this is related clinically to a condition that's virtually universal, presbyopia, or age-related farsightedness. As we get older, everything gets stiffer, right? But one of the things that gets stiffer is the lens. So this kind of soft, stretchy, transparent material becomes less easily deformable. And it gets harder for it to change shape when you focus on something close. And what happens is that when you focus on something close, instead of the lens relaxing into this more globular shape, it stays flatter, and it's much harder to focus on things that are nearby, which is why we need, as we get older, most people in their late 30s, early 40s end up needing reading glasses, which are positive diopter glasses, um, convex glasses that are trying to help us focus the written word, these small things that we're trying to look at, the needle that we're trying to thread, um, when our lens is unable to change shape to focus. So let's watch this video and allow it to teach us a little bit.
Presbyopia develops when our eye's natural lens loses its flexibility. Focusing up close becomes more difficult. The eye's ciliary muscle contracts, which causes our flexible lens to thicken or change shape. This change in shape allows us to see objects up close. As we age, our lens becomes less flexible and resists changing shape, so our ability to see up close is diminished. Usually, the first sign of presbyopia is the need to hold a newspaper or other reading material at arm's length to be able to read them.
I want to talk now about the correctable causes of vision problems, the things that people wear glasses for, just so that you have a better understanding of what they mean. So, what we call as lay people farsightedness is called medically hyperopia, and it occurs when the eyeball is short, is not so deep, and the light rays that are focused by the cornea and lens focus on a point behind the retina. And you correct this with a magnifying lens, a convex lens, a positive diopter lens, that brings that focal point a little bit forward, allowing focus at the proper place. Myopia, what we call nearsightedness as lay people, happens when the eyeball is too long or too deep, so that the focal point of the light of the image that is being focused by the cornea and lens is actually in front of the retina, and it is corrected by lenses that are concave, um, negative diopter lenses that then allow that image to be a little bit uh, smaller, but to focus on the proper focal point a little bit back from the one that is natural. And so emetropia is normal vision. And with emetropia, you can see we have our cornea, the lenses in here. Much of the focal power, much of the what happens in terms of focusing light occurs in the cornea, and the lens it just assists it. So in emetropia, you have your image coming in and focusing properly on the retina. With myopia or nearsightedness, the image focuses inside the eyeball, in front of the retina, and it is corrected with concave negative diopter lenses that allow the image to focus in the proper place. With hyperopia, also called hypermetropia or farsightedness, the image is focused, whoops, back here behind the eyeball, and we use a convex lens um, to bring that focus to the proper place. This is a retina. It says as seen through an ophthalmoscope, and you'll learn very soon in H&P that that's not exactly true. When you're looking through an ophthalmoscope, you see about the diameter of the optic disc. So you can look at any part of the retina, you can see the fovea, you can see vessels, or you can see the optic disc, but you don't see all of this. This is all seen through a camera. So when the eye doctor takes a picture of your retina, they're getting a picture that looks something like this. You can see the choroid, which is the vascular tissue behind the retina, and that's where all these vessels are. So I've always said that the eye is like a little window through which you can actually see blood vessels in the body, and you have arteries and veins. Um, they converge at the optic nerve because if you remember, the vessels traveling to the eye, into the eye, and out from the eye travel through the optic nerve. And again, this is the fovea, which is the center of the macula. The fovea is a little bright spot in the center of this area that is a little bit depressed. That is the point in the eye where the greatest detail is seen. The optic nerve has no neuroretinal cells covering it, which means the optic nerve is where your blind spot is. Okay. When you look at something, you are focusing both eyes on the same image, and the image ends up inverted and reversed on the retina. So think about when you take a magnifying lens, which is a biconcave lens, and use it to focus an image on a piece of paper. Have you ever done that? Well, you get an image that is inverted and reversed left to right. So here we have this arrow with a purple end and a blue end, and on this eyeball, that image is upside down and left to right. And in this eyeball, the image is also upside down and left to right. We can think of each eye of the retina as having a temporal and a nasal half, a temporal and a nasal half. And you can see here that on the, let's say we're looking down, are we looking down from the top? We're probably looking down from the top here, which makes this the left eye and this the right eye. That the temporal half of the retina of the left eye is receiving the tip of the arrow, and the nasal half of the right eye is receiving the tip of the arrow. Okay, and those fibers from that image are coming back into the optic nerve, and we're showing in this picture the kind of the left half of the visual field, which is the right part of the image, on this temporal retina and on this nasal retina. Those fibers end up traveling together through the optic tract. So here you have the optic nerve. Here you have the crossover of the nasal fields through the optic chiasm. Okay, so the images cross over at the optic chiasm, and then they travel back into the brain through the optic tract. And we're going to come back to this when we talk about field cuts. But just sort of thinking about how an image is projected, both the light image onto the retina, and then subsequently the information, how it is carried back into the brain. Okay. Now, I said that the retina is made up of neurons, and it is made up of a number of kinds of neurons. The neurons that are deepest are the light detectors, the rods and the cones. And they are in direct contact with something called the pigment cells of the choroid, which is sort of an epithelium on this very vascular membrane that is the middle part of the structure of the eyeball. And that's where they're getting their nutrition from. So we have rods and cones. And then they are communicating with neurons that are called bipolar cells and horizontal cells. And those neurons are doing some processing and communicating with cells that we call ganglion cells. And the ganglion cells then are sending their axons to the optic nerve. So I'm not going to make you memorize the retinal layers, but I want you to understand the essential structure, that you have this choroid, which is a pigmented epithelium, in contact with the rods and the cones, which are the light detectors and detect light at various wavelengths. And if you're looking at a section under a microscope, you see nuclei there, which is why it's called the outer nuclear layer. And it's outer because it's the closest to the choroid, to the outside of the eyeball. Okay? And then the inner nuclear layer are these cells that are processing things, beginning to take information from the light detectors and detect things like movement and edge. And then they are in contact with ganglion cells, which are collecting all that information and sending it through their axons into the central nervous system. You might also want to notice that there are glial cells that are in here supporting everything. And here we have the micrograph where you can see your choroid, which is the vascular layer with a pigmented epithelium, the rods and cones with the outer nuclear layer, the inner nuclear layer, and then the ganglion cells, and then these axons. So the light actually has to get through all of these cells to make it to the rods and cones. At the fovea, um, the layers are thicker and the outer layers are thinner, and the cones and rods are denser. This again is based on a micrograph. So there's something called Nissl substance, which is in cell bodies. So you can here see the cell bodies of the rods and cones, and you can see that there's just a ton of rods and cones here. And then the bipolar cells and ganglion cells are off to the side. So this is the part of the eye where we just get the greatest focus, the greatest intensity of the image. So let's talk about the photoreceptors. Um, they are a little different structurally, and they function slightly differently. They all have something called an outer segment in which you have these sort of layers, which are the light detectors themselves. An inner segment and a cell body, and then sort of a very short axon that synapses with the bipolar cells. And rods have their photopigment, and this is a rod. They have a lot of a photopigment called rhodopsin, and rhodopsin breaks down to a very wide bandwidth of light, and it doesn't help us detect color. It just detects light. It's very sensitive to light. There's a bit more of it than we see in the cones. Um, and so there's more photopigment, it's more sensitive photopigment, and it's less specific concerning bandwidth. So rods are very sensitive to light, and they don't need a lot of light to fire. In the peripheral retina, you have almost entirely rods. So they are extremely sensitive to light, but they're not great for, they are completely insensitive to color, and they're not great for acuity. But rods are what help you see at night. If you don't have rods, you can't see at night. And also peripheral vision. The cones, there are different types of cones that have slightly different photopigments, each of which breaks down to a limited bandwidth of light. And that means that the photopigments in the cones are color sensitive. Um, they are somewhat less sensitive to light. There is less photopigment in the cone than there is a rod, and so they need high illumination levels. When you're lying in bed at night and the light is very dim, you really don't see color very well. And in fact, you tend to see in grays, and it's only as light levels increase that color begins to come back into your vision. There are few cones in the periphery, and there are a ton of cones in the fovea. And so the fovea is responsible for vision when there are highlight levels. They allow you to see color, and they are responsible for the very high visual acuity of what you are focusing on, and which is focused in the fovea of the retina. Okay. And this is kind of a nice little slide. So it's showing the fovea, and right at the very very tip of the fovea, we don't have any rods and we have a ton of cones. Okay, so these are cones and these are rods. All right, so you have rods out in the periphery and you have a ton of cones in the middle. You have a few cones moving out. You have absolutely no rods right at the fovea. The optic disc does not have photoreceptors, so that's our blind spot. And this is just sort of saying what we saw in that previous slide. So we already mentioned that the rods have a lot more segments. They're bigger, they're taller, and they have more photopigment, and that photopigment is achromatic. It's a wide bandwidth of light, doesn't help you distinguish between colors. Um, the cones are a little shorter, they're cone shaped, they have less photopigment, they are adapted those pigments to color. Um, you have lots of them at the fovea and few of them in the periphery. You have lots of these in the periphery. Your visual acuity is best in the fovea. Your light sensitivity is best in the periphery. And the optic disc is where the blind spot is. So let's talk a little bit more about those cones. There are, as in most individuals, there are three kinds of cones. And when people have a genetic defect where they either have an absence of some of those cones or they have cones that don't function properly because of a genetic change, you can have color blindness. And the most common kind of color blindness is an X-linked absence of L cones, which are detecting light in the red and yellow bandwidths. It could also be green cones, but the most common is this X-linked absence of L cones, of red-yellow cones. And because it's X-linked, it occurs in men primarily. Mothers, women are carriers, and the only women who are protanopes, who have this red-green color blindness, are women who've inherited a faulty X chromosome from both parents so that they have a complete absence of the L cones. And it removes the long wavelengths, the red primarily. Um, so it shortens the visual spectrum, and these people have a really hard time distinguishing between red and green, seeing red. And I'm just going to tell you a little story. My son, okay, so my grandma Boo Hecker, my paternal grandmother was actually colorblind. She had a lot of serious eye issues, but that was one of them. And so she only had one child, and her child was a son. And sort of by definition, if she had two abnormal X chromosome genes for L cones, he inherited an abnormal gene. So my dad was colorblind. I'm not colorblind, but apparently my son, I inherited uh, clearly the faulty X chromosome from my dad. Um, I didn't inherit a faulty X chromosome from my mom because it's not all that common, and so I'm not colorblind. But my son Johnny is. And Johnny decided when he was in high school that he wanted to be in the Coast Guard. And in fact, he wanted to go to the Coast Guard Academy. And he worked his tail off in high school. And I remember saying, Johnny, what I read is that they're pretty fussy about color blindness. And he said, I'm not too worried about it, Mom. And he worked his ass off and he actually managed to get a Coast Guard Academy appointment, which is hard. There were only, I'm not sure now, but 250 cadets accepted a year, and they did not accept people for congressional appointments in the way that West Point and the Naval Academy do. So he got accepted and he got sent off for a Department of Defense physical at some office in Durham that contracted with the Department of Defense. And when he came back, he said he passed his physical. And I said, really? Did they do a color blindness test? And he said, yeah, Ma, they used that little Ishihara book like you have in the office, which should have given me a clue. But Johnny ended up getting inducted into the Coast Guard at the beginning of the summer. The cadets go in June, I think. So he graduated from high school, had a couple weeks off, and I took him to New London and dropped him off. And he was inducted, and at the end of the day, they send them all out to march around a little bit, and parents can wave, but you can't talk to them, and then you leave. So I left, and I went to my brother's house in New Jersey for a few days. And a couple days later, my cell phone rings, and it's my son. And he says, Ma, they're kicking my ass out. And my first thought was, what did you do? But I didn't say that. I said, why? And he told me that they had redone the physicals on everybody and they had done a different color blindness test, which is called the lantern test, where they flash red and green lights of different intensities and you have to identify the color. And he failed that, and he and about a dozen of his classmates were being sent home for various reasons having to do with their physicals. Um, in retrospect, I'm pretty sure my son memorized the Ishihara book because he had access to something most people don't. But the Coast Guard has no tolerance for color blindness because they say that the ability to detect only faintly discernible differences in color are extremely important for search and rescue. So, Johnny, fortunately, we had paid a deposit at state, and he ended up going to state very happily. I'm not sure that it was, I know it was a terrible disappointment at the time, but I think in retrospect, his life worked out okay. This slide is showing the different kinds of cones. So people say blue, green, and red. Um, they obviously overlap, which is what allows us to have a wide range of the ability to detect different colors. There are apparently a small percentage of females who have a fourth type of cone that's living in here somewhere, and that may expand their ability to see different gradations of color in these wavelengths. And then rods have a very wide range of wavelengths. And this is that Ishihara test. So I don't know if any of you have ever seen these books, but if you are colorblind, the differences between the reds and greens are such that you cannot read these numbers. So it's hard to reproduce on the computer screen. Um, I can see a 5 here. I can see a 6 here. I can see a 2 here. My son can't. And there are some other kinds of color blindness having to do with the blue and yellows, again, because of abnormalities in the gene uh, coding for that particular photopigment. I'm going to talk a little bit more about vision. The processing of visual information is extremely complex, not completely understood, although there's an awful lot of research that's gone on in the last 20 years or so about this. And it starts in the retina. So when I said that those bipolar cells and horizontal cells are beginning to process visual information, um, that's interesting, right? Because we tend to think about processing as happening in the brain, but within the retina, we're already beginning to process information about contrast, about edges, and perhaps about movement. And the neurotransmitter that is used by those cells in the retina is glutamate. And then the retinal ganglion cells, again, are continuing to process information, and here it's about shape and movement primarily. And then those axons take that information, which includes not the raw information from the rods and cones, but the somewhat processed information from the bipolar horizontal cells and the ganglion cells. And then those ganglion cells generate action potentials, which send signals through the axons to the through the optic nerve and back into the central nervous system. There are a number of retinal diseases that can cause trouble with vision. People who have vitamin A deficiency quit producing photopigments and can actually develop permanent blindness if it's profound vitamin A deficiency. But the earliest symptom is poor night vision because of a lack of adequate photopigments in rods. So poor vision and low illumination. There is an inherited disorder called retinitis pigmentosa, in which over time there's a failure of the ability to maintain the receptor cells, the photo cells, and it leads to blindness. A common cause of blindness, in fact, the leading cause of blindness in old people, is age-related macular degeneration. And there are two types. There's something called dry macular degeneration and wet macular degeneration. And in dry macular degeneration, you just have a lot of cells proliferating in the macula, which begin to obscure that very detailed vision in the macula. And in wet macular degeneration, the invasion is of capillaries, and so people lose central detailed vision. And initially, they often will complain of difficulty reading. They may have some blurred vision. The wet form of macular degeneration is treated by laser surgery. So laser pulses are used to kind of close up the capillaries, but it does destroy some normal retinal cells. And the dry form of macular degeneration is treated with injections of uh, biologically engineered substance that decreases the growth factor problem that is causing the proliferation of cells. Sometimes the retina can detach. The retina is adherent to the pigmented epithelium of the choroid, but sometimes it pulls away from the choroid. And if the neural cells pull away from the choroid, they're unable to get oxygen and they essentially die. And this is treated by, again, using lasers that kind of weld, they cause a little bit of scarring, and they weld the neural layer of the retina back to the pigmented layer of the choroid. Um, but again, you get some blind areas because you destroy some neural cells with a laser. And then finally, diabetics develop a number of eye problems that together are called diabetic retinopathy. They develop little aneurysms which sometimes burst and cause hemorrhages, and the bleeding into the choroid damages the rods and cones and the neurons in the retina and results in blindness in the areas that are affected. And again, lasers can be used to kind of seal up these little aneurysms, seal up um, leaking blood vessels, but they also damage the surrounding neural tissue, but they can prevent further damage and blindness. And now we're coming back to the visual pathways. And this is stuff from that's going to come up again and again and again. And this may be your first exposure to it, or you may have been exposed to it as an undergrad. But this is stuff you've got to know. So remember that the axons of the retinal ganglion cells, and these are afferent axons, they form the optic nerve. Okay, and the image is reversed and upside down. So what is in front of me on my right side is being projected upside down onto the left side of my retina. And what's on my left side is being projected upside down on the right side of my right retina. Okay. And in addition, it's what's over here is being seen on the nasal side of my right eye as well as the temporal side of my left eye. And what's over here is being seen on the nasal side of my left eye as well as the temporal side of my right eye. So the fibers from the nasal side, they cross over, they decussate in the optic chiasm. The optic chiasm is a nerve tract that is right under the pituitary gland. So we'll come back and talk about that a little bit later, but that's important to know as an anatomic relationship. So we have the optic nerve coming back out of the orbit into the skull, and then they join at the chiasm, and the temporal fibers just keep going back. So temporal fibers are seeing the vision from the other side, right? And they're going directly back. And the nasal fibers, and they're seeing the vision from the other side, cross over. And so we end up with what's going back on the right side of the brain is the left side of the visual field. And what's going back to the left side of the brain is the right side of the visual field. There then is, after we have the optic nerve, we have the chiasm where the nasal sides cross, and then we have the optic tract carrying the contralateral information or visual information. In the brain, they terminate the optic tract in various nuclei that are related to vision, and particularly the ones that help us actually see terminate in the thalamus in something called the lateral geniculate body. So remember, the thalamus is the relay station, the diencephalon. And then from the thalamus, fibers that are called optic radiations. So there are cell bodies here. There are cell bodies here, ganglion cells, sending these axons, synapsing in the lateral geniculate body of the thalamus, and then sending out fibers that are called the optic radiation. Again, the information from the left visual fields end up in the right occipital cortex and vice versa. So let's think about what happens if we cut one of these pathways. And this is actually, this picture is kind of nice because it shows some other relationships. For instance, it shows that some of those fibers are going to other nuclei. So there's an importance of the light levels telling the hypothalamus something, because the hypothalamus is involved in regulating circadian rhythms. Um, the light levels also have to travel to the Edinger-Westphal nucleus because of the light reflex of the pupil. They also travel to something called the superior colliculus because eye movements and head movements have to be coordinated. So there's stuff going on in there to help coordinate how all these muscles work together. But the primary visual information is going to the thalamus, to the lateral geniculate nucleus, and synapsing there. All right, so here let's talk about visual fields. And again, remembering that the field, that the picture itself is inverted. If you sever the optic nerve, you lose the vision from this eye. Okay? If you sever or do something to damage the optic chiasm, what are you going to lose? You're going to lose the information from the nasal halves of the retinas. And the nasal halves of the retinas are seeing the outside visual field. And so you're only going to see the image that formed on the temporal halves of the retinas. And that's going to be what's in here. Okay? Because the image is flipped and inverted. If you were to sever the optic tract, you would lose the information from the temporal half of the left retina, the nasal half of the right retina, which means you'd lose everything that you would see on the right side. If you were to sever this tract, you'd lose everything that you would see on the left side. So these are different kinds of field cuts that can result in visual problems when there is damage to the optic nerve, to the optic chiasm, to the optic tract, or to the optic radiations, these fibers coming out of the lateral geniculate body, or to the visual cortex. Okay. And here we have an illustration showing a lesion of the right optic nerve causes blindness in the right eye. A lesion at the optic chiasm causes something we'll call hemianopsia. We're losing half of our vision, and it is temporal hemianopsia. We're only seeing what's in the middle, and that has to do with a lack of decussation or crossing over of the information from the nasal retina in both eyes. Okay, so even though it's the nasal retina, because of the image being upside down and backwards, what we're seeing here is what ended up on the temporal retinas, which is the central visual fields. If we cut one of the optic tracts, we're going to get a different kind of hemianopsia. And in this case, we cut the right optic tract, and we will get a hemianopsia in which we only see the information that is coming from the left side. So you will see actually coming from, yeah, from the right side. Sorry. Is that right? Yeah, because the left side is getting right information, and we have cut the right optic tract. If you cut or damage, say by a stroke, part of the optic radiations, you can get a quadrant-synopsia. If there is damage to the striate cortex, you can get a hemianopsia with central sparing. These are the three I care about, A, B, and C. Okay, and here we have some different kinds of causes of those sorts of lesions. So for instance, and again, you remember that what is internal to the visual field gets projected to the temporal side of the retina. That what gets projected to the nasal side of the retina is what crosses over. Okay, what gets projected to the temporal side of the retina is the middle. Okay, so if we have a lesion such as a tumor or inflammation from a problem like multiple sclerosis or syphilis that can cause optic neuritis, or we have ischemia to the optic nerve, or we have very high levels of intracranial pressure from a condition called idiopathic intracranial hypertension that is compressing a single optic nerve, you will have blindness on one side. That's your visual field defect, is that you can't see out of your right eye. You can only see out of your left eye. Problems at the optic chiasm often occur because of masses near the sella turcica. You could have an aneurysm, a swelling in the pituitary. There are benign-ish tumors called adenomas that can make the pituitary enlarge and put pressure on the optic chiasm. There are tumors of the meninges called meningiomas, and there is a kind of tumor called a craniopharyngioma that can put pressure on or cause destruction to the optic chiasm. And when that happens, you only see what's the part of your vision that is making it to the outside of your retinas, which is the inside of your visual fields. So you get bitemporal hemianopsia. So you see the middle and you don't see peripherally. Okay? If you cut the optic tract, you get a one-sided hemianopsia. So you only see the information, depending on the tract that's cut, from the information that's making it to the occipital cortex on the other side. So if you cut the right optic tract, you will see the information that's making it to the left optic cortex, which is the right field of vision. Okay, and again, these are additional problems like tumors, Strokes, infection, inflammation can cause problems in the optic radiations or in the actual um, cortex, occipital visual cortex, that can cause different kinds of field cuts, like quadrant field cuts. So here we have a couple of clinical cases. We have a 30-year-old woman who comes in complaining of headaches and loss of peripheral vision. So you do a good exam and you check something called visual fields, and you determine that she has bitemporal hemianopsia. So she seems to have sort of tunnel vision. She can see the center of her vision, but she can't see peripherally on either side. So where do you think the lesion is? And in this case, that lesion is in the optic chiasm. And the most common cause of a lesion in the optic chiasm is a pituitary adenoma, often a prolactinoma. And we'll talk about the pituitary gland as we move a little further into the course. But that's just something that could happen. Um, you may have a 67-year-old who's complaining of a gradual loss of vision in his right eye and difficulty walking. And when you do your visual acuity exam, he seems to have no vision in his right eye. He's blind in his right eye. He also has an odd gait. He has some weakness in his lower extremities, and he may have um, some sensory problems. And this is somebody actually that I have seen who, when we did the workup, including an RPR, a rapid reagent, or rapid plasma reagent, which is a syphilis screening test, it was positive, and the syphilis screening test reflexed to a specific uh, test called FTA, or fluorescent treponemal antibodies, which was positive. And this gentleman turned out to have neurosyphilis. So he had optic neuritis, inflammation of the right optic nerve that led to blindness, as well as a number of other neurological problems that can be associated with syphilis. He had to be treated with IV penicillin. He did not regain his vision, and he had dementia in addition to these other issues. So he went on to have a lot of difficulties from tertiary syphilis, neurosyphilis, probably that had been around for 20 years or so.